Welcome back to the DNA replication video, where we will talk about specifically how DNA replicates as the cell is actively going to be growing and dividing. Okay, and so as I mentioned in the other video, it gets rather content heavy, but there are some tricks that we can kind of just think it through and things will make sense. Some of the names get kind of complicated, I admit, but you can do it nice and easy, and it won't be too bad. All right, so 5' to 3' is what we want to remember. A polymerase, a DNA polymerase specifically, can only polymerize DNA in one direction using that 5' carbon end and pairing it with 3 carbon end of the next nucleotide. Okay, using a triphosphate nucleotide. So 5' to 3' is the only way it can do that at all. It's the only way. It does not go from the 3' to the 5'. The polymerase just did not evolve that way. It found that 5' to 3' was energetically favorable using 2 ATP leaving groups, or 2 triphosphate leaving groups, and it stuck with it, and that's how life evolved afterward, right? So 5' to 3' is the only way that it works, but that runs into some questions because we have an anti-parallel structure, right? The anti-parallel structure of DNA lends itself that there would only be one active 5' to 3' end as we go along down the fork. As we pull it apart, we're going to pull apart 2 cords wrapped around each other, and one side is the only way we can go, and we're going to have to go backwards on the other side. Okay? So we're pulling apart 2 cords that are wrapped together, 2 extension cords that have been stuck together for all Christmas, and you're taking them apart, and they're all wrapped together. Okay? Only one of those ways is how the DNA polymerase 3 can actually work. Okay? It goes 5' to 3'. Okay? You can see it here. You can see it here. Okay? And that DNA polymerase 3 has already synthesized this as I'm pulling it apart. Okay? Pulling it apart, and it is replicating. Okay? So what do we do with the other side? Okay? That side we can kind of tell by me saying it is not 5' to 3'. Instead, it is 3' to 5', right? See, 3', 5'. Okay? So what we need to do is lay some track down on the other side, called an RNA primase, or the RNA primase puts down the RNA primer that we need. That's the track that we need. Okay, now we have our track that is going in the opposite direction, going in the 5' to 3' direction. Remember, the opposite direction of the fork. Okay, on the lagging strand is what we call this, the lagging strand. All right, and the lagging strand now has a pathway to go 5' to 3'. DNA polymerase 3 finds that RNA primer, and it says, hey, there's a 5' to 3' end here. Let's do this. It's going to catalyze that reaction until it hits another RNA primer. It will leave a gap. It will leave this small gap. That phosphate group will not have paired over to the next one and next nucleotide. And so we need a DNA ligase to repair it. Okay. So without even thinking about it, we've kind of already talked about a few things. We talked about DNA polymerase 3 running along doing its 5' to 3' action. Both the leading strand and the lagging strand. Just does so on the lagging strand, making these Okazaki fragments. Fragments of the RNA primer plus the DNA copy until it runs into itself and creates that nick. Those are the Okazaki fragments. Okay. The RNA primase we talked about also. Right. Lay down that track. And we talked about DNA ligase repairing those nicks. Okay, so we already kind of talked about a few things. We need the track layer, we need the actual doer, the polymerase, and we need our DNA ligase. Okay, but we also need to get into how we get rid of that RNA primer on our DNA. So we're going to need another polymerase that does that. We're still polymerizing DNA together, so we still need to call it a DNA polymerase. Why is it called 1? 1 was discovered first, named, and ended up doing this one. Okay. The second one was named. DNA polymerase 3 was now found to do the action. Okay. Only after the third discovery. Okay. So DNA polymerase 1 removes the RNA primer in order to make the gap for DNA ligase to come in. Now, as I'm pulling it back, I'm using my own energy and my own arms to pull the two cords apart, right? But what does that in DNA? It is the helicase. The helicase is going to unwind helix. Unwind the helix, helicase, right? Enzyme that unwinds the helix. So now we're still making some sense here, right? We got most of our enzymes named, and we got most of our list named over there already. Okay. The only other thing that we need to talk about is how we're going to end this thing, and how, if we kept doing that to an extension cord, right? Two extension cords, or the one wrapped around itself, it's going to tangle. It's going to get really tangled at the other end toward you, right? And so we need to unwind that tension, and we're going to use topoisomerase in order to relieve that tension. It'll intentionally apply a nick, cutting the cord to allow the tension to come, and then DNA strand. The DNA ligase will again repair that. All right. And so the only other thing on this list that I did not mention is the single-stranded binding protein, which is going to help unwind this fork. Okay. And so as I pull it back, as we are getting that tension there, that is actually called a fork, like a fork in the road kind of a fork. And we make a lot of these forks all the way down the line. And so we put in the DNA polymerase 2, DNA polymerase 3, helicase, ligase, primase, sliding clamp, topoisomerase, sliding single-strand protein, and then topoisomerase again. Okay, introduce a single break, chromosomes, real-seq DNA. Okay, and we have DNA polymerase 2 with the repair function. Okay, that is going to be how we are going to repair minor mutations, as we call proofreading. Okay, that's what the DNA does when we are repairing it. It's proofreading it. And we talked about replication forks. Okay, we're not just going to have one, we're going to have multiple of them. Okay, and those make bubbles, and those replication bubbles spread out and join together enzymatically favorably to two new strands. Right? If we did it at just one spot, it would take forever. And as I mentioned, we in lab we have telomerases. Okay? Telomerases are where I think we age from. Telomerases are going to end DNA replication. It's going to add on some DNA bases to allow the DNA to not skip what needs to be written. Okay. And Elizabeth Blackburn here, as you see, is the one who discovered these telomerases and is credited with the discovery. So keep her back in mind that she discovered telomerases. She goes pretty synonymously with them. Here are some differences you can look over about the origin of replication. In prokaryotes, for instance, it's only one, versus the eukaryotes, which has an abundance of DNA, needs the multiple, otherwise it would take forever. The rate of replication, though, in prokaryotes is 10 times, maybe 20 times as fast as eukaryotes. DNA polymerase types, 5, and we only talked about 3, but look how many there are. There's 14. What do they all do? They must do something with mutations, proofreading, being redundant with DNA polymerase 3 sometimes. A lot of different things can happen. Okay. Telomerase, the bacteria do not need it because they can undergo what's known as rolling circle replication, which I'll get into in the next slide. RNA primer removal, both of them have that RNA primer for the lagging strand. Okay. Strand elongation, note we are calling DNA polymerases the 1 and 3 only in the prokaryotes, and they have these Greek letters in the eukaryotes. That means we aren't really studying those. Okay. We are instead studying the bacterial prokaryote ones. Okay. Keep those in mind. Finally, ending this video, this is rolling circle replication and an illustration of how DNA is replicated in prokaryotes rather than eukaryotes. So, okay, it goes around in a circle, and so you wouldn't need the telomerases. You wouldn't need multiple replication forks because it's a lot smaller and more succinct. All right, and that concludes our DNA replication lectures. We'll see you in RNA transcription.